Today we're going to be taking a look at and talking about muscle contraction, from the release of acetylcholine at the neuromuscular junction to excitation-contraction coupling to sliding filament theory and ending with muscle relaxation. There are three different types of muscle, all with different functions: cardiac muscle, smooth muscle, and skeletal muscle. For the sake of this video, we will be looking exclusively at skeletal muscle. Skeletal muscle is made up of bundles called fasciculi, which house the individual muscle fibers. Muscle fibers are covered by an outer membrane known as the plasmalemma, which is part of a larger unit referred to as the sarcolemma. Within the sarcolemma are the aforementioned muscle fibers. Here's a picture to help better understand. Muscle fibers are made up of myofibrils, which are even smaller fibers composed of basic contractile elements of skeletal muscle. In the space between myofibrils is a gelatin-like substance known as the sarcoplasm. The sarcoplasm houses an extensive network of transverse tubules, which are extensions of the sarcolemma. These interconnected T-tubes run along the myofibrils, allowing nerve impulses from the sarcolemma to rapidly reach individual myofibrils. This network of tubules is known as the sarcoplasmic reticulum. The sarcoplasmic reticulum stores calcium, which is essential for muscle contraction. Now, before we go any further, we need to take a closer look at myofibrils. Each myofibril can be broken down into repeating segments called sarcomeres. Sarcomeres are the most basic functional unit of striated skeletal muscle. Sarcomeres are made up of two protein filaments, actin and myosin. The end of each myosin strand is folded into a globular head called the myosin head. These heads interact with specialized sites on actin filaments during muscle contraction, in a process known as sliding filament theory. Thin actin filaments are composed of three different protein molecules: troponin, tropomyosin, and actin. Each actin filament contains sites for myosin heads to bind to at intersectional cross bridges. Let's dive a little deeper into the sliding filament theory. When myosin head cross bridges are activated, they bind with actin, resulting in a structural change in the cross bridge, which causes the myosin head to drag the actin filament toward the center of the sarcomere. This act of dragging is referred to as power stroke. Here's a picture to help us better visualize everything we're talking about. As you can see, the sarcomere is joined from end to end at what are known as the Z-discs. As you can see, each sarcomere contains an H-zone, an M-line, an I-band, the Z-discs that we already talked about, and an A-band, along with the myosin and actin filaments. The pulling of the thin actin filament past the thick myosin filament shortens the sarcomere and generates force. But this energy comes at a price, and it all starts with alpha neurons. Alpha neurons are nerve cells that innervate muscle cells. The process that triggers a muscle fiber to contract is known as the excitation-contraction coupling. This process begins with an action potential, a nerve impulse from the brain or spinal cord that reaches an alpha motor neuron. The action potential travels down the neuron's axon to the axon terminal. When the action potential arrives at the axon terminals, the nerves release a neurotransmitter called acetylcholine. Acetylcholine is released from docked vesicles via exocytosis. Acetylcholine diffuses across the synaptic cleft, binding to receptors on the sarcolemma of the muscle fiber. This causes an increase in permeability of sodium ligand-gated ion channels, moving sodium into the muscle cell in a process called depolarization. Acetylcholine is rapidly broken down in the postsynaptic cleft into acetic acid and choline by the enzyme acetylcholinesterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles. This process is known as reuptake. So, back to depolarization. If the cell is sufficiently depolarized, action potentials will then stimulate the opening of voltage-gated sodium channels in the T-tubules of the muscle cell. these channels are physically linked to calcium channels in the sarcoplasmic reticulum, and they open as well. Released calcium then binds to troponin, changing its shape and moving the tropomyosin molecules off the myosin binding sites on the actin molecules. Once the tropomyosin has been moved from these sites, the myosin heads can attach to the actin, forming a cross bridge. Now, muscle contraction requires energy. Myosin heads contain a binding site for the molecule ATP, adenosine triphosphate. An enzyme on the myosin head called ATPase splits the ATP to yield adenosine diphosphate, ADP, and inorganic phosphate, IP, as well as energy. The energy from the breakdown of ATP causes the myosin head crossbridge to pull the actin filaments inwards. This occurs along the entire length of every myofibril in the muscle cell, contracting the muscle. When an ATP molecule binds to the myosin head, the myosin detaches, breaking that cross bridge. As ATP continues to break down, the myosin head again attaches further along the actin filament, repeating the process. This repeated pulling is the sliding filament theory. Muscle contraction will continue for as long as there is calcium available. At the end of the muscle contraction, calcium is then pumped back into the sarcoplasmic reticulum. Muscle contraction ends when neural activity ceases at the neuromuscular junction. Calcium is then returned to the sarcoplasmic reticulum via an active calcium pumping system, one which also requires ATP. Thus, energy is required for both contraction and relaxation of the muscle. So let's recap. The sequence that begins with a nerve impulse and ends with muscle contraction is called the excitation-contraction coupling. An action potential travels to the neuromuscular junction where it releases acetylcholine, opening ion gates on the muscle cell membrane. This causes sodium to depolarize the cell. If the cell is depolarized enough, it will then generate another action potential. The action potential then travels into the T-tubulus system, causing stored calcium to be released. Calcium ions bind with the troponin. This binding changes the troponin-tropomyosin complex to change shape and move into a groove on the actin. This then exposes active sites on the actin filament, allowing binding to the myosin heads. Myosin heads bind to ATP, which is split by ATPase into ADP and P. The energy from this breakdown causes the myosin head to tilt, pulling the thin filament past the thick filament. This is a power stroke. ATP molecules again bind to the myosin head, causing it to detach from the actin. But when the ADP is broken down, the myosin head reattaches to the actin. This repeating process is, again, sliding filament theory. Muscle contraction ends when activity ceases at the neuromuscular junction. Calcium is actively pumped back into the sarcoplasmic reticulum for storage. Tropomyosin moves to cover active binding sites on actin molecules, leading to relaxation between myosin heads and the binding sites. The muscle relaxes and the excitation-contraction coupling process is complete.